Welcome to my channel, and today's topic is DNA replication. Okay, so what is replication? Replication is the process by which new DNA strand is formed from the parental DNA. And here the two terms very important, that is the denaturation and the renaturation. What is the denaturation? The denaturation is the breaking of the two double-stranded DNA into the single strand. Okay, two single strands. And the renaturation is the process where the single-stranded DNA that transforms into the double-stranded DNA. Okay, the two processes are very important for the replication. And there are many enzymes. Actually, the replication is a process that is found in eukaryotes and also the prokaryotes. Okay, and different types of enzymes that are very important for the completion of the replication. And here first we discuss about the enzymes that are very important for the completion or for total completing or helping to complete the replication. First we just learn about the enzymes that are related to the replication, and the second we discuss about the process. The process generally if we just divide it into three steps, the first one is the initiation, second one is the elongation, and the third part, that is the termination. Okay, and another part is also including is here, that is proofreading. Okay, now we first learn about the enzymes that are related to replication. Okay. Now we just learn about the enzymes. First one is the DNA protein. Second one is the DNA-C protein. Third one is the DNA helicases. And fourth one is the Ssb protein. These are the proteins, and another proteins that are also involved for the replication. We learn after learning of this, we then learn about that enzymes. Okay, so first one is the DNA protein. Actually, DNA protein is responsible for the localized strand separation at ODC. What is the ODC? ODC is a place where the initiation of the replication starts. Okay, this is known as the ODC. So, what is the role of the DNA protein? DNA protein that is responsible for localized strand. Actually, it helps to localize the strand separation at ODC. Actually, it identifies the ODC. Okay, it identifies the ODC and forming the single-stranded DNA. Okay, this is the DNA protein that actually helps in the replication. And ATP is needed here. That means energy is obviously needed for completing this process. Second one is the DNA-C protein. It helps to join the initiation complex and contributes to formation of two bidirectional replication form. What is bidirectional? In case of replication, it is not unidirectional. Actually, after initiation, it bidirectional means the process continuing in the bidirectionally, not unidirectionally or in one way. Okay, it continues in the two ways. Okay, or in the two direction, and that's why it is known as the bidirectional process. So this is the DNA-C protein. DNA-C protein actually join the initiation complex. That complex actually help for the initiation of the replication. Okay, so it helps to join the initiation complex and contributes to formation of two bidirectional replication form. It helps to form the two replication fork. Okay, clear? This is the role of DNA-C protein. Third one is the DNA helicases. What is the role of the DNA helicases? It unowns the double helix using energy derived. Okay, first we just learned it unowns the double helix. Unwound means this is double helix. So unwound means just double helix changes to single stranded DNA. Clear? And that means this is the role of the helicases. So helicases helps to unwound the double helix, and it is also here ATP. The energy is needed for doing this work. The next one is the Ssb protein. Ssb protein actually it keeps the unwound DNA strand in an extended form. Actually, its role is very important. Ssb protein. Helicase just help the double helix to make it single strand. Okay, but Ssb protein, that is the single stranded binding protein, it binds. Why? Because it helps to continue or to remain the DNA helix into the unwound situation or unwound situation to the extended form. Clear? So what is the role of Ssb protein? It keeps the unwound DNA strand in an extended form. So these are the role. So first one, DNA protein. What is the role? Responsible for localized strand separation at ODC. Just localize the strand which one is the ODC, and this is the place where the initiation starts. Clear? DNA C protein, that is join the initiation complex. Okay, the first identified the initiation place and then initiation complex is added here or bind there for doing the replication. Third one, helicase, it helps to the double stranded DNA into the single stranded DNA. That means it helps to unwound the double stranded DNA to the single stranded form. The fourth one, the Ssb single stranded binding protein. What is its role? Its role to remain unwound situation. That means the helicase that helps to unwound the helix, and its role, it keeps the unwound DNA strand in an extended form. Okay, so these are the role of the 4 enzymes. Another enzymes are also involved here. We learn after that. Okay, next one is the DNA topoisomerase. Topoisomerase, its important role is, this is responsible for removing the supercoils in the helix. The DNA coil, that is in case of double helix or double stranded situation, the supercoil is formed. That means coiling is very highly like that. Okay. Supercoil. This is called the supercoil. Coiling is very tightly packed. That is known as the supercoil. So its function is what? DNA topoisomerase that helps or responsible for removing the supercoils. It removing the supercoils into the relaxed situation. Okay. It has 2 type of topoisomerase. One type 1 and the type 2. Type 1 actually reversibly cut a single stranded of DNA. It reversibly cut the single stranded DNA. And second, its function is, it has 2 functions. That means nucleus activity and the ligase activity. What is the nucleus activity? That means cutting activity. Clear? And the ligase activity, that means adhesive. It acts as adhesive. Okay. That means cutting and joining. Both capacity they have. Okay. The second one is the type 2. And okay. And here the ATP is not required. That means energy is not needed here. The type 2 there, that binds tightly and makes transient breaks in both strands. It helps to cut the both strand. Transient break, the both strand, not the single strand. It helps to cut the, actually it has the same activity, cutting activity, but the type 1 activity is different, or type 1 and type 2, the 2 topoisomerases have the different types of activity. Cutting and adding activity is same, but how these active or these function in that situation. Okay, like that type 1 we learned here. Type 1 that single-stranded DNA, reversibly cut the single-stranded DNA. But type 2 that have the activity or have the capacity to cut the double-stranded DNA. Okay, and here the DNA xylase is one kind of topoisomerase that is invented in E. coli. Okay, invented in E. coli. It helps the negative supercoils into the relaxed circular DNA. So what we learned here, topoisomerase, the topoisomerase actually we learned here that is responsible for cutting the double-stranded DNA. Okay, but the two types of topoisomerase we learned here, we know here that type 1 that helps to cut the single-stranded DNA, but type 2 that has the capacity to cut the double-stranded DNA, and both strands they actually cut the two strands simultaneously in a transient way. Okay, and here the ATP is not needed, but in case of type 2 the ATP is very important. Another topoisomerase that is xylase, we know that is actually found in E. coli, and its role is it helps to relax the double-stranded DNA, but the negative supercoils. Okay, negative supercoils means actually when the strand after relaxing the supercoil into the single strand, it has a chance to make again to form the supercoil. Okay, so that is controlled. Okay, that is controlled by the xylase. Clear? The primase. What is the role of the primase? It synthesizes the short stretches of the RNA that are complementary and antiparallel to DNA. Its function actually the function of primase is to synthesize the RNA. Okay, to synthesize the RNA that is antiparallel and complementary to the DNA. Clear? So these are the few enzymes we learned here, topoisomerase and the primase. Another enzymes are very important, that is DNA polymerase. Okay, DNA polymerase and the ligase. Now we learn about that. Okay, now we learn about the activity of the DNA polymerase or DNA poly. The three types of DNA polymerase are involved in the replication process. First one is type 3, DNA pol 3. Second one is the type 2, DNA pol 2, and the last one is the type 1. Okay, type 3, the role of the type 3 is actually it helps in the elongation process. We learned the three steps of the DNA replication. One is initiation, second one is the elongation, and the last one is the termination. Okay, so here actually the role of the DNA pol 3, it helps in the elongation process by adding the nucleotides using the RNA primer, the 3' hydroxyl group. Okay, this is 3' hydroxyl group of the RNA primer. Actually it just takes the 3' hydroxyl group of RNA primer as the initiation part. Okay, and using this part, it added the nucleotides with the template DNA. It begins to add the nucleotides along the single-stranded DNA using the 3' hydroxyl group of the RNA primer. Okay, so here which one is the template DNA? That means double-stranded DNA that breaks into the single-stranded DNA. Clear? And this one single strand, each one acts as the template. Okay, and by using this template, the DNA pol 3, it added the nucleotides of the new DNA strand. Okay, and here we just learned it when the replication process we learned. So here with the template DNA, the DNA pol 3, it added the nucleotides using the 3' hydroxyl group of the RNA primer. RNA primer is very important. If RNA primer is not there, the replication process is never continuing. Okay, so So RNA primer is very important, and initiation part, the replication starts at the initiation portion. There RNA primer is must be needed. Okay. And the second one, DNA Pol 2. It's another activity is there. It helps in proofreading. What is proofreading? Like if during the replication, or actually we just follow the elongation process where the nucleotides are added. Okay. So if any nucleotide that is mismatched to the template, okay, or that is not necessary for the new DNA strand, so this mismatched nucleotide is replaced, okay, by the correct or the accurate nucleotide. Okay, so this is the proofreading process, and this process is done by the DNA Pol 3 obviously, and it is done according to 3' to 5' activity. But that means actually the replication process, you know that it actually occurs from the 5' to 3' direction, but the exonucleus activity that occurs in 3' to 5' direction, in the opposite way. Actually the replication process is continuing, but checking is also continuing simultaneously. Okay, and if during the process of exonucleus activity, this is done by the 3' to 5' direction. Clear? Okay, the type 2. Type 2 is what? It helps to repair the DNA damages. DNA Pol 2, its role, the actually repair, it helps in the damaging part of the DNA. And a type 1, it synthesizes DNA like that, the DNA Pol 3, and second 3' to 5' exonucleus activity, and the third one also, it removes the RNA primers. That means during the exonucleus activity, when it removes the mismatched portion or mismatched nucleotide from the DNA strand, it also helps to remove the RNA primers, because RNA primer is not necessary when the replication of the strand is completed. Okay, so it also helps to remove the RNA primers. Clear? So these are the three type of the DNA polymerase we learned here. First one is the type 3. It has the exonucleus activity from the 3' to 5' direction, and also it added the nucleotides. That means it helps in the elongation process. Okay, and the second one is the DNA Pol 2. It helps also the repairing process. Okay, and the type 1, it generally works like that. DNA Pol 3 synthesizes DNA, and exonucleus activity, except activity is that it helps to remove the RNA primers. Okay, so these are the activity of the DNA polymerase. Okay, now we learn about the ligase. You know that ligase is actually the helps to ligase. The function is the ligase enzyme. Actually, it helps, or it adding, or its activity just like is adhesive activity. That means it if there is two fragments of the DNA part, or the DNA nucleotides or DNA strands is there, so ligase activity is that to attach the two fragments with each other. Actually, it helps generally, very important in Okazaki fragments. We learn about the Okazaki fragment. What is the Okazaki fragments? So what is the activity of the ligase? Its activity like that, the adhesive activity. It helps to join the fragments of the DNA strands. Clear? So these are the enzymes that are very important for the replication. So first one, we just summarize the again. First one, DNA A protein, and the second one, DNA C protein. Third one, helicases, single-stranded binding protein, Topoisomerase, primase, DNA polymerase, and the ligase. And another enzymes that are also important for the replication, we just learned, but these are not very so much important, necessary for continuing the replication, but actually main important role we have seen here. Okay, now another accessory accessory enzymes, you can say that accessory enzymes or helping enzymes that are also important for doing the replication. We just learned here, obviously as a list. Okay. These are the another enzymes. First, type 3, Pol 3, DNA Pol 3, and under DNA Pol 3, two enzymes are very important: DNA E alpha and epsilon DNA Q, and it helps in polymerization and it helps in 3' to 5' editing. Okay, and RNase H, RNHA, it helps. That means it removes the RNA primers, and under these are the enzymes. Actually, where are they? They remain, or some functions also. So, Hol E present in core, DNA beta, DNA N sliding clamp, and tau DNA X joins leading and lagging DNA Pol 3, DNA and DNA Pol 3. That means it forms to make organize or form or organize complex, to make the complex. And tau star, again DNA X, that also binds the clamp loads and Ssb and clamp, it binds clamp loads and Ssb. Okay, and delta Hol A, clamp loading, Hol B, same function, clamp loading, Hol E binds Ssb, helps to bind the Ssb, and Hol D, that also it binds the Ssb. So these are the another enzymes that are also involved in the replication process. Now we learn about some terms that are very important before going to the replication process. What are these terms? First one is origin of replication, or the ORIC, replicator, replication bubble, template strands, replication fork, bidirectional replication. Okay, these are the terms. First we learn about this term, so then it is very easy to understand the process of replication. Clear? Okay, these are the terms that are related to replication. First one is the replicator. What is replicator? The initiation of the replication is directed by a DNA sequence. Okay, that is called the replicator. Okay, initiation of replication is directed by DNA sequence. Okay, the replications will be occurred in which direction, that is totally dependent on the DNA sequences. Clear? And that is called the replicator. Second one is the ORIC. I just first I just explain this topic or this part, this term. Before that, here I also again explain you. ORIC is the starting point. Starting point means the specific region where DNA double helix denatures. Okay, where the DNA double helix denatures. That means the replication or the initiation process starts. The point that is known as the ORIC. Third one is the replication bubble. What is the replication bubble? The denatured segment of DNA. You can see that this is the double helix DNA. This is again double helix DNA. That means double strands are there. And here you can see the one strand is here, another strand is here. That means here a bubble is formed. Okay. So replication start this way or this way. Clear? So this is the replication bubble. Actually, where the joining is relaxed, or the two strands, the coiling of the two strands that relax into the single strand formation, this is called the bubble, replication bubble. Next one is the template. Which one is called the template? This you can see this part. This is one strand, this is another strand. These two strands when remain singly, then each this strand and this strand both act like the template. That means according to this, the replication starts, and according to this, replication starts. Okay, so this is template and this is template. Clear? Next one is the replication fork. Replication, which one is the replication fork? So you just see here the bubble formation, clear? So this process, this bubble formation when extends, that means this is the starting point, or the by forming the bubble, it shows that replication is possible here, okay? And after that, this shape is formed, okay? This shape is formed, and this Y-shaped structure is known as the replication fork. Clear? And the last one is the bidirectional process. Bidirectional process means in case of circular DNA, obviously circular DNA, the replication process starts from here. I just, okay, no, like that, okay. So here the replication process in this way and in this way, clear? So this is the bidirectional process, not here the replications process occur in the linear way, okay? It occurs in the two directions, and that's why it is known as the bidirectional process. Clear? These are the terms related to the replication. We just in this video we learn about the enzymes and different terms that are related to replication, and in my next video we learn about the replication process. Clear?